Hello, everybody. So first of all, thank you very much for the opportunity to be here and to speak to you all. So I'm Yonit, and I'm actually currently a postdoc at the Physics of Life Cluster of Excellence of the TU Dresden in Germany and the MPI-CBG. But the work I'm going to tell you about today is from my previous institute in the group of Professor Kinneret Keren. And so what I'll be talking about is the mechanics of Hydra regeneration and the role of topological defects. Okay, so just an outline what I'll be covering today. So first I'll talk about our slightly earlier work on the nematic organization and the topological defects in the supracellular actin-5 organization in regenerating Hydra, which was mentioned today already, so thank you. And then I'll be talking about our slightly more recent work on mechanical strain focusing at the defect sites and its role in the formation of the head. And then I probably won't have time, but maybe just give a teaser on some older experiments, but a new paper on the effect of confinement in modulating the body plan of regenerating Hydra. Okay, so the question that we are kind of trying to address in this work is regarding morphogenesis, so the emergence of functional patterns in living organisms. And it's really not well understood how these robust and functional patterns emerge in the face of a very noisy environment. And so generally, morphogenesis emerges from the integration of multiple biophysical and biochemical processes with extensive feedback between them. And, whoops, that's not what I meant to do. Interesting, okay. But it's really not well understood how these processes are coordinated across scales to form robust and functional outcomes. And for this reason, we think it can be very useful to try and kind of use a coarse-grained approach and to try and form an effective description of the process. And this requires identifying the relevant coarse-grained variables and then using them to form a predictive framework. And so generally, research in the field tends to focus on the importance of biochemical factors in patterning. But it's becoming increasingly apparent that mechanics can play a powerful role in pattern formation and especially in coordinating it across scales. And so our aim is, by focusing on mechanics, to try and develop a coarse-grained understanding of pattern formation and its dynamics. And so for this, we take kind of two main approaches. So one is focusing on actin and myosin, which are sort of universal force-generating system in biology. And the other is actually by applying mechanical perturbations. Okay. So for this work, we use the Hydra as a model system. So the Hydra is a small predatory freshwater animal. It's about 1 centimeter long, and it has a very simple body plan. So the body is formed of a hollow tube with a foot on the one end and a head on the other. The head is surrounded by tentacles that it uses to catch prey. And this hollow body tube is comprised of 2 cell layers, which I'll return to in a moment. First of all, why is the Hydra such a great system to study these questions? Well, it has remarkable regeneration capabilities. So you can cut a small piece of tissue from the body of the Hydra, and it will always first fold into actually a hollow spheroid, and within roughly 2 days, it will regenerate completely into a fully functional mature animal. So you can see a movie of how this looks. And what you can appreciate here is how dynamic this process actually is. So this is a tissue spheroid, and you can see it's expanding, contracting, moving around a lot, changing shape. And here, within less than 2 days, you already see the emergence of the tentacles, and you have a fully functional adult animal. So returning to these 2 cell layers that comprise the body of the Hydra, these 2 layers function both as an epithelium, a skin, and also as muscle. And as such, they contain a very striking array of muscle fibers. So these are actin and myosin fibers. And so in the outer layer, this is what you see here in green. They're aligned really nicely, as you see, parallel to each other and along the body axis. And in the inner layer, that's what you see in purple, they're again aligned very regularly parallel to each other, but circumferentially to the body axis. And this alignment is nematic, so although the animal has polarity, a head and a tail, the fibers do not. And indeed, we can observe nematic topological defects in the fiber organization during regeneration. So generally, during regeneration, we see three types of defects. So these are the +½ the -½ and the +1. And so just to maybe clear what you're seeing here. So above, you're seeing images of the actin fibers in regenerating hydrospheroids, and below is the quantification extracted from these images. So the black lines represent the orientation of the fibers, and the color is the nematic order parameter. Okay, so if we look at mature Hydra, it turns out that the location of topological defects is not at all random. It's very regular. It will be the same between all animals you look at. And the location of defects coincide with the morphological features. So the Hydra has a +1 defect at the mouth. That's what you see up here, +1 defect at the foot, and then also an additional +1 defect at the tip of each tentacle, which comes with a pair of -½ defects at the base of each tentacle, leading to, if you sum up the charges of all these defects, topological charge of +2, which is what you would expect of something that is topologically a spheroid. Now, interestingly, the Hydra can sometimes regenerate with multiple body axes, so an extra head or extra foot. And in this case, you have an extra +1 defect. And again, this comes balanced out with a pair of -½ defects at the junction between the body axes. And so up to now, I was showing you images from this outer layer, but it's the same story in the inner layer. So the defects are located at the same locations. The only difference being that now, where the fibers are circumferential, the +1 defects are this vortex shape rather than the aster shape. Okay, so what happens during regeneration? So the first thing to know is that when we excise a piece of tissue from the body of the Hydra, initially the alignment of the fibers is maintained. So you have an open piece of tissue with a perfect array of nematic fibers. And the next step is that this open piece of tissue falls into a spheroid. And the resulting spheroid contains a large region in which the alignment of the fibers is maintained, but it also contains a region that loses its supracellular fiber organization, and this is where the edges of the tissue kind of stretch to meet and seal the spheroid. But the way that these are organized is, again, highly stereotypical and reproducible from sample to sample. And so I'll try and explain to you how this organization looks. So the ordered array spans roughly 2/3 or 3/4, if you like, of the circumference of the spheroid. And then the disordered region will be located at the so if you like, the top and the bottom of the spheroid, with also a bridge connecting it. And what this means is that the top and the bottom, which lack nematic organization, can be identified, in fact, as foci of the nematic organization, because they're mostly surrounded by a radial organization of fibers. And so what happens next is that over roughly 24 hours, nematic fiber organization begins to appear also in these initially disordered regions. And now defects appear, and the defects appear exactly at these early foci that we identify in the organization of the fibers. So these are the regions marked in these schematics with the yellow and the blue dots. So we've seen that initially, when you cut a piece of tissue, the array of the fibers is maintained. And then the folding introduces defects. And these defects are long-lived, so we can actually follow them over the whole regeneration process. And we've seen that the defects in the mature animal correspond to the locations of the morphological features. So this led us to ask, can we use this to sort of develop an effective description of defect dynamics and learn from it something about the morphogenesis process? Okay, so in order to do this, we wanted to follow the evolution of the defect configuration over time. So what we did is we counted the different types of defects that we saw over regeneration, over multiple samples. And so the results are summarized in the plot you see below here. And so the first 24 hours are this phase of induction of order. So you start with no defects because you have an open piece of tissue, and then just a spheroid with a disordered domain. And then defects begin to appear. And at this point where the whole spheroid is covered with a nematic array, by far the most common type of defect we see are these +½ defects. Those are represented here in blue. And then the number of +½ defects actually begins to decline, and the number of +1 defect increases until finally you reach the configuration typical of a mature animal with 2 +1 defects of the head and the foot. And here I'll note that I'm actually ignoring the defects associated with the tentacles. And the reason for this is that the tentacles actually tend to appear later on, and after the defects that define the body axis, the head and the foot are already there. Okay, so what is happening to these +½ defects? So by far the most common thing we see is that pairs of +½ defects actually come together and merge into +1 defects. In addition, we also observe annihilation of +½ and -½ pairs. And it turns out that the formation of the head and the foot is actually directly related to the defect configuration and to an asymmetry in the defect configuration that emerges at these two early foci. So typically what we see is that in the two foci, one of them will form a +1 defect, and the other one will form a pair of +½ defects. And if we follow these defects now through regeneration, we see that at the +1 defects, these will almost always become the site of the mouth of the regenerated animal, whereas the pair of +½ defects that come together will form the site of the foot of the regenerated animal. And in fact, we can even follow the cells that are located at this site of the defect by inserting a photoactivatable dye and then labeling selectively a group of cells. And we can see that at these +1 defects, it's in fact the same group of cells that is located at the site of the defect and will become the site of the head. Okay, so an interesting question that remains here is how is this asymmetry in the defect configuration, which is related to the polarity of the animal, related to the polarity of the original animal from which the tissue was taken? And so this was work of, yeah?
Just a short point question on the previous thing you said, that you can track the cells by marking them, and so the cells that have a +1 defect or are near a +1 defect become the head?
Yeah.
And is the same also true if you track the cells that are initially near +1/2 defects? Do they come together and make the foot? Is it the same cells, or do the defects move relative to the tissue?
So, so it's not the same. It's not the same group of cells with the +halves. If you like, you can think about it as the +half actually moving sort of in relation to the cells. But generally, the region here where the +half defects are coming together, so you can see it kind of here, right? The green dots. So that's kind of the center of the group of cells. So before it's located sort of between the two, and later they'll kind of come together there. But generally, there's a lot more cell movement in that region compared to the +1.
So that's the same animal we see just later, on, like in both. It's the same animal.
These two, yeah.
Yeah. Cool.
Mm-hmm.
Thank you.
Yeah. Okay. So returning to here, this was work done by another former PhD student in the group. And so what she did is she labeled this, the tissue with two different colors, one marking the originally head-facing side and one marking the originally foot-facing side. And what we saw from these experiments is that actually the polarity can't be trivially inherited here, because there's actually a region in which the originally head-facing side and the originally foot-facing side come together when this spheroid seals. But still, it turns out that there's a strong memory of polarity. So the +1 defect, which will become the site of the head, always forms within the originally head-facing side, whereas the pair of +half defects always forms within the originally foot-facing side. Okay, so I'd like to just summarize what I've told you so far. So we've seen that the nematic field and the defects kind of provide us with a nice, effective description of what is happening here during regeneration. So the nematic provides us with this coarse-grained, slowly varying field, where the location of the organization centers that will become the head and the foot coincides with the defects in this field. And we've seen also that the location of the head and the foot can actually be identified from the nematic pattern from the very onset of regeneration as these foci of the actin fiber organization, even before the point defects have formed. And the asymmetry in the defect configuration reflects a memory of polarity. Okay, so this led us to kind of the next obvious question, which is, well, okay, so what can be special about the defect sites? And in order to kind of try and answer this question, we wanted to see whether there's a relation between the actin fiber organization and the defects and tissue dynamics and mechanics. And so what we needed for this is, in addition to... Oh, yeah.
One more question. Is there any way in which you could independently control the location of the defects? Is there, for example, anything like an optogenetic tool or something that you could use to really see whether you can move the defect and then the anatomical structure will move? Is there a way to do this?
Yeah. So it's very challenging. First of all, due to, if you like, the topological constraint, right? You can't completely get rid of defects. There has been recent work actually making the Hydra into a torus, and then you can do that. I can tell you about it later. But but in terms of, you know, if you're trying to perturb fiber organization, etc., you're always perturbing other stuff. So we've had to approach this indirectly. Again, I'll get to some stuff later on. Yeah.
Can I ask another question before you move on? You talk about moving defects, but these actin fibers presumably are regenerating. So is there motion of physical objects, or are these patterns that move?
Okay, so there's not a perfect understanding of this yet, but so these fibers are located within the cells, okay? And the reason that they span across the animal, that they're supracellular, is actually that, if you like, sort of protrusions holding these fibers between cells are kind of attached to each other, and that's how they form these very long lines. So in order for a defect to move, you actually need to remodel these connections, right? And this does happen, but it happens slowly. So there's not, I don't think it's correct to think about it. Okay, and sorry, in addition to that, you do have some cell neighbor exchanges. So if you think about that, that will also help you organize your fibers. So things are moving, they're mostly remodeling, and it's happening slowly. Okay.
Can I ask a question, please? What is the role of cell division? That's surely going on here. You presumably could track it. Is it correlated with defects, etc.?
So, first of all, strictly cell division is not required for regeneration. So you can actually regenerate if you block cell division, although it's a bit impaired. We've looked into this, although not in great detail. So there seems to be a bias in terms of the relative direction, if you like. So when cells divide, right, there'll be an axis over which they separate. And there's a small correlation between that and the fiber orientation. We haven't seen any relation to defects. So whether there's more cell division at defects or not, I can't tell you for sure, but we haven't seen something yet. Okay, so in order to be able to try and correlate the fiber organization and the location of the defects with tissue dynamics, we need a way to actually visualize what is happening to the tissue. And so to do that, we, for various technical reasons, actually needed to do quite a lot of computational image analysis here and basically separate the signal from this actin, which marks also the fibers and also the apical junctions of the cells. And by doing that, we could visualize separately the actin fibers and also the cell outlines. And now, if I show you this movie, you can, I think, better appreciate kind of what is happening to the tissue in terms of deformation, etc. And then we also wanted to do this quantitatively, so we need to segment the cells and also correct for the geometry, because I'll remind you that this whole thing is actually a very curved surface. Okay, so
What's the left and right there?
and so on the left are the fibers, and on the right are the outlines of the cells. Oh, yeah. Okay. And so now that we have this possibility, yes.
Okay. Does the substrate affect the motion of the defects on the organism, like whether the friction with the substrate?
Okay, so we have these two cell layers, and in between them, yes, there's an extracellular matrix, which, is that what you mean by the substrate?
Yeah.
I don't know the answer, whether it affects and what role it plays. There's very little work on that, but it's an interesting point. Yeah. Okay, so now that we can do this, so I'll just draw your attention before I play the movie to this site that I'm marking with this yellow circle, which is this early focus of the actin fibers, which will become the +1 defect. And if you look at this movie now, what you will be able to see is that this site undergoes these multiple large transient deformation events. So the cells at the early focus and later defect site become transiently really stretched. You can see this quantified in the images below. So below, what you can see on the left is a map where the color code is the cell area, and on the right is the cell anisotropy. And again, I'll actually return here to the beginning of the movie, so again, you can look where we've marked with the green frame is when these events are happening. So the cells at the defect site are transiently really stretching and then going back to their original size, and this is happening at the early foci and later on when these become the defect sites. So you can see in these images an example of such an event. So again, on the left, what you're seeing are the fiber organization and cell outlines just before the event. And then on the right is what happens during the event. So you can appreciate this really large change. And this is at a +1 defect site. If we look at what is happening at a pair of +½ defect sites, so you do see some local deformation, but it's a lot less pronounced than at the +1. Okay, so we wanted to quantify this deformation and both see how it is localized in space, but also how its transient nature in time. And so what I'm showing you here are plots that are that represent the area of mean cell area over time during regeneration, dividing the tissue into 3 different regions. So the orange are the cells closest to the defect, and then green is further away, and blue is far away. And here we use actually the topological distance of the cells from the defect, because of course absolute distance is changing dramatically over this time. So you can see by kind of these very tall peaks in the orange graph above how the cells at the defect region are transiently stretching, but then the baseline is really very similar. And this is for the future head site, but if you look at the future foot site, you have smaller events, yes, but a lot less significant. Okay, so we also wanted to look how the strain or the deformation looks as a function of the distance from the defects in more detail. So what we did is we took all these events and looked at the deformation as a function of the distance, and again comparing what is above is the future head site with below the future foot site. And what I'd actually like to emphasize here is that, so once the point defects are formed, there's actually a difference in the fiber organization between the future head site and the future foot. So the +1 defects are undergoing these more dramatic events, but this difference is actually apparent even before we can see the point defects form and the difference in terms of the defects. And just to say that even more dramatic than these local stretching events is that, so over regeneration, the hydro tissue undergoes cycles of osmotic swelling and then some ruptures which release pressure. And if we look where the ruptures are happening, they are happening predominantly, or exclusively, at these early foci / later on point defects in the fiber organization. And even more dramatically, occasionally we get these enormous holes forming. And somehow the tissue still reseals and manages to regenerate. But this is just for entertainment. We don't actually understand very well what is happening. So kind of to summarize these findings, what we've seen is that really from the onset of regeneration, the cells that are located at these early foci of the fiber organization, and specifically at the future head, are experiencing really a unique yet transient mechanical cues. So if we look just at these large stretching events and actually count how many events are happening over regeneration and plot the cumulative number of events. This is what you can see on the left. We see really this increase, so multiple events happening again and again at this future head compared to the future foot. But then what you can see in the plot on the right is that again, the transient nature of these events. So really this difference between the head and the future head and the future foot, but mainly between the actin foci and the rest of the tissue, is not apparent if you just average the areas over the whole regeneration process. So really only if you look only at the these stretching events, then you see the difference. Okay, so first of all, we wanted to understand, to see whether we could understand this observed deformation pattern and its relation to the fiber organization. And so for this, what you need to appreciate is that the hydra tissue is essentially a muscle. And what this means is that its activation is actually calcium dependent. And this means that if you want to think of an active stress that is associated with the pneumatic, we think that the right way to think of it is as something that is not only anisotropic, according to the direction of the pneumatic, but also changing in time, and also time dependent. And again, just to enjoy some more movies, but really to see here how different, if you want to think of the stresses that that this tissue is experiencing, how dynamic and different and time dependent they are. So we wanted to see if we could actually sort of quantitatively understand the cell deformation pattern as a function of the fiber organization. And for this, we've had a collaboration with Marko Popovic and student Suganthan from MPI-PKS in Dresden. And so they've built a model for the hydro tissue mechanics. So the tissue is modeled as an elastic deformable shell. And this shell is now coupled to a pneumatic field with an active stress that is proportional to the pneumatic, but also has transient activation. And then we also have a hydrostatic skeleton holding the shape. Okay, so the model is realized using a generalization of a vertex model, but to a 2D closed surface. And so the energy of the vertex model includes the sort of more familiar terms. So these are the area elasticity, the bond tension, and the perimeter elasticity. We also include the surface bending energy because we're not considering the 3D, but this thing has a thickness, so it can't bend too much. But then what we have in addition is this pneumatic that is assigned to each cell, and the active stress over here, which is proportional to the pneumatic, but has transient activation dynamics. And so then we can look at how the cell deformation pattern looks when you impose this transient activation of the fiber organization. So we're looking here at one spheroid from 3 different views. So you see the +1 and the +12, and you can see that when we activate this stress, then you have this large deformation at the defect. Okay, but this doesn't tell us yet about sort of the functional role of the defect in morphogenesis. And in this regard, what is important to know is that the group of cells located at the head of the hydra so at the head of the hydra, there's a group of cells that is called the head organizer. And this group of cells is responsible basically for generating the signals that are required for maintaining the head, the structure of the head. And so in a mature hydra, this is continually maintained, but during regeneration, you actually have to form this group of cells anew. And what we know is that, of course, in the mature hydra, this group of cells, this head organizer, is located at the site of the +1 defect. Okay, so what do we know actually about this? We know that the group of cells that is located finally at, that forms the head organizer and is finally located at this +1 defect is in fact, from the beginning, the same group of cells that is located at the early, one of the foci of the fiber organization, and which then forms the +1 defect, and throughout regeneration is undergoing these large recurring stretching events. And so this leads us to propose this feedback mechanism for head formation. So the part that I've shown you today is the fact that we have these defects in the fiber organization, and these basically work to focus the strain in this location and undergo these large strain events. And what we'd like to propose is that in response to this strain, you actually have a mechanosensitive process which is leading to production of a morphogen or molecule that is required for head formation. And that then in response to that, the fibers tend to align with the gradient of the morphogen. And this is something that was proposed as well by Friedhoff and Christina recently, which leads to a reinforcement of, again, the strain focusing, leading to positive feedback. And experimentally, we can investigate this by trying to perturb the molecules that are involved in the head formation. So we can do this in two different ways. One is using a specific drug. It's not so important, but in this case, what we see is that actually fibers don't even form in the originally disordered region. And of course, there's no regeneration. And we can do this another way by using a media of high osmolarity, which has been shown to interfere with this signaling pathway. And what we see in this case is kind of nice, that sometimes +1 defects form, but they quickly unbind into pairs of +½ defects. So you actually end up with spheroids with 4 +½ defects, and they don't regenerate. Okay, so this very briefly, but in the model, we can actually investigate the feasibility of this feedback fully by including an additional field, the morphogen field, and then the feedback manifests in the fact that the morphogen field has a production term that is strain dependent, and then the nematic has a term that tends to align it with the direction of the gradient, and from this, we can get, and then now that we have this model, we can actually look what happens if you start from a spheroid which has initially no morphogen and has the fiber organization similar to what we see in the experiments. And so what you can see in this movie is now that you start having these transient stretching events, how also the fibers align and you get the configuration of defects, and also you get the localization of the morphogen at the defect sites. So this is really kind of shows us that this kind of feedback could work to give you self-organization of a head organizer. Okay, so to summarize, I'll just leave you with this idea of the nematic organization of the actin fibers as this mechanical morphogen. So it's a field that varies in space and time, and defects in this field provide local mechanical cues. And our hypothesis that we're suggesting is that the organizer formation occurs as a self-organized dynamical process through a closed-loop feedback that promotes the localized stress enhancement at the +1 defect sites. And really, this identifies the defect sites as key locations at which the integration of all these multiple processes is likely to be taking place. And so I said that this would be just a teaser, so if you're interested in what happens when you now try and put these tissues in narrow channels, you can ask me about this later, so let me just skip. And yeah, so I'd like to thank my previous group, and specifically Professor Kinaret Keren and our collaborators, Marco and Suganthan. And just again, a very quick note. So now I've started my postdoc in Dresden, and I'm working a little bit on mechanical organization in gastroloids, but predominantly on robustness of morphogenesis in zebrafish tailbud. Thank you.
I'm sure there's a lot of questions. Yeah, there it is. Okay, so I start. Oh, I guess you're the only one of us. Okay, we'll go one here, and then maybe you, and then we continue.
Yeah, thank you very much. Very, very interesting. There was one aspect that didn't play a role in at least I didn't see it. But so the fact that we're two systems of active stresses on, but separated by the thickness. Okay, so this is like an active bilayer. Yeah. And so, okay. If you understand my question, I mean, I was surprised. So it would have an impact on the model, right? So there would be spontaneous curvature as well as area elasticity with a preferred area. But probably, you know, it didn't play a role. So is it irrelevant? I mean, this beautiful feature of having two orthogonal stress directions separated by thickness, does it play a role in your system?
So I think, I mean, the short answer is, again, we don't yet know. It's certainly fascinating, and I think there are some clues where we're not capturing everything, because I think the inner layer could be playing a role. In fact, actually, when I showed you this giant hole, what happens is also that the inner layer and the outer layer kind of peel off each other, and actually I think that the inner layer is kind of helping maintain the shape a little bit. But again, I'm not sure. So it's definitely there, and it's definitely interesting to understand what the role is. I think what I can say is that in the context of what we've been looking at, we found that we don't need it in order to explain what we're seeing. So whether the two are just following each other, I don't know. So yeah, it's very interesting, but we haven't got there. Yeah.
I guess, yes.
Yeah. Do the defects help make the morphodynamics more robust to noise in the system?
Can you repeat that?
Do the defects actually help the morphodynamics become more robust to noise?
So this is what we're suggesting. Again, I can't say anything for sure, but in this picture that we're suggesting, exactly the fact that you have the defects of the specific organization of the fibers, if it is coupled with the morphogen production, the fact that you would have a positive feedback would help you robustly form your head there. Okay? But again, for us, this is at the level of a proposed mechanism. Yeah.
Thanks.
Could you, I didn't really get that, and explain again what it is that decides that one defect becomes a +1 defect, and whereas the other one remains a pair of +1 halves?
Yeah.
What sort of decides which one does what?
So what we do know is that they form according to this memory of polarity. So but this is quite coarse. So one of them will form within the region that was originally closer to the head, and the other one closer to the foot, but not at the edges. And again, I'm talking about a pretty rough localization. So there must be something that is holding this memory, whether it's gradient, whether it's mechanical properties, it could be many things. And the way we think about it is that within these regions, specifically where the defects will form is in these actually really foci of the fiber organization, and this is what is helping reinforce it. But I'll emphasize that our model currently does not capture that. So the model currently doesn't capture the difference between the two.
And then they sort of, as soon as one has become more a +1 defect than the other, there's some sort of repression going on where there's more strain at the +1 than at the other one, and then the wind signaling sort of helps decide. Is that what is going on?
Maybe. I mean, yes, like this sounds like a likely scenario, but again, it's not something that we know. Yeah. Yeah.
Yeah, I'm wondering about the elongation, like how does the spheroid become something elongated and develop tentacles? I mean, does your model of thinking provide any insight into that?
So it's a really good question, and it's something that we've been looking at on the side a bit. It's in a way, again, counterintuitive, because if you think of these fibers, right, contracting in that direction, then you would think that, well, it could be contracting in the other direction. What I can say is that you have some cell rearrangements. So this is helping you elongate. You also actually have kind of a change in the resting state, if you'd like to call it, of the cells. So maybe you saw in the images I showed you, the cells were kind of isotropic, but in mature Hydra, they're super stretched in the direction of the body. And this happens kind of suddenly when they move from being like more. So it's probably something to do with the sort of baseline of muscle activation. But again, this is things that we really don't know. It's just, preliminary.
Maybe Silke very fast.
Just a little clarification. So I didn't really understand what your morphogen does mechanically. Does it enhance pneumatic contractility or reshape it, and then it pulls and opens and closes the hole, or did I understand this correctly? So what we're assuming is that somehow the fiber orientation is just
sensitive to the gradient, so it's helping direct, if you like, where the fibers are aligning. Yeah.
And that creates, and then morphogen is created.
So through this picture of the feedback, then if the fibers are more radially aligned, this region will experience more strain because you have more pulling, and then that will increase the in turn the morphogen production.
Okay, yeah, thanks.
Hi, great stuff, really interesting. So I had two questions actually relating to the active pneumatic sort of component of that. You described the morphogens and how they evolve and all these things, which are very interesting. But from like the active pneumatic on a curved surface type of, we know that curvature and topological defects kind of interact, right? So the stretching could be explained by essentially this type of mechanism. But what I wanted to ask was about the +1/2 defects. Do they form and then move, or do they form? Because it is known also that +1/2 defects are motile.
Yeah.
But the +1 is not. So it could be that it's,
Yeah. I mean? So all I can say in this context is that, yeah, our results match what you would expect in that context, so in the context of the motility. Whether this is by chance or whether there's actually the same physics going on, I don't know. But yes, they do move.
But
and the +1s do not.
I see.
Yeah.
All right, thank you.
Right, what's your first time? Oh, okay. Yeah. You were first. I'm not sure who was first.
All right, cool. Thanks, awesome stuff. And I'm going to be annoying and keep asking questions for which there are no yet known answers. So I get that, but I was super excited, of course, like many others, about this strain-dependent morphogen production as well as morphogen concentration-dependent alignment. And so perhaps those mechanisms are not known here, but could you talk about at least how much is known about these mechanisms in other, maybe, more classical organisms?
Yeah. So, okay. So in terms of having production that is strain-dependent or stress-dependent, so there are some kind of molecular pathways that are known to be mechanosensitive. The most famous is this YAP-TAZ. So actually in the experiments, like in the cell monolayers, they were able to show that at defect locations you have less or more, depending on the defect, crowding, so more or less stress, and actually in response you have a difference in the signaling, which affects, in that case, cell proliferation or cell death. So this is one example, but there's not a lot of examples, certainly not from development, so yeah, still open, yeah.
And then for the other one, the morphogen density-dependent alignment?
So in that context, again, I mean, so we have mainly experiments from Hydra where you can put things that either enhance this wind locally, or as I told you, the perturbations, and you kind of see that the fibers will tend to align. Like if you put a peak, like really strong peak of wind, you will align the fibers to there, and similarly the other way around. So this is the evidence that I know. Yeah.
Pretty much our time, but we'll make two quick questions. So
yeah, I mean, my question is actually related to this point. So you showed this very interesting result that if you inhibit wind with ICRT14, then you get no alignment or very little alignment of the fiber. That's surprising. I mean, I was always wondering in the first place how they align.
Yeah.
I mean, if you think about an imagine, it's some steric interaction, but these can actively remodel, so it's not obvious at all why they would make this sort of supracellular fibers to begin with. And so now it looks like you have a perturbation. Did I understand correctly that perturbation already sort of inhibits these long supracellular fibers?
Exactly. So it allows you to maintain the fibers where they still exist.
Yeah.
But yeah, you don't form new supracellular fibers in the disordered regions. I think though what I can say is that, so these different perturbations are perturbing slightly different parts of the pathway. And there's probably something a bit more subtle going on in like which parts of the pathway do what in terms, and which interact with the alignment, because even what you saw is that, so using this drug, we get no alignment in the disordered regions, whereas when we use the osmolarity, we do get alignment, but the +1s are not stable. So yeah, really cool, there's probably more to get into in that respect. Yeah. Okay.
Super short.
Yeah. Hi. So my question is, like, when you cut something, then you always start from a spheroid. So does it fold on itself before you start studying it? Like, how do you get to a spheroid?
Yeah, so it always falls into a spheroid, and actually we have earlier work where we explored different geometries of tissue. So they all fall into a spheroid, but if you think about it, then the configuration of the fibers will be different in the different spheroids. And actually this maps kind of nicely to the formation of the defects and also the body plan. So by getting kind of crazy shapes at the beginning, you can get multiple defects and multiple body axes. So, yeah.
All right, let's thank Yonit again.